In this chapter, we're going to look at ectodermal placodes and epidermis. Ectodermal placodes are thickening of the surface ectoderm. They form rudimentary structures from which organs can develop. The cranially sensory placodes include the olfactory, lens, and auditory placodes, and they give rise to the most peripheral neurons of the head that are involved with senses and balance. If you look at the picture that's there in the blue box, here we can see where the cranial placodes are during very early development. During later development, we can now see the actual locations of these cranial sensory placodes. Now, in addition to cranially sensory placodes, we also have other types of placodes which give rise to hair, teeth, mammary, and sweat glands. Now, different placodes arise from a common panplacodal field, and the panplacodal field is shown in the region that is there in the blue box. The panplacodal field is competent to form different placodes when induction occurs by neighboring tissues. Now, the default state of the panplacodal field is to form lens tissue. However, the neighboring tissues will provide signals that will cause the panplacodal field to differentiate into different types of placodes like the lens placode, the auditory placode, or the olfactory placode, etc., etc. The portion that is shown in the blue box actually shows all the different factors that are involved in forming the panplacodal field. So we start with the preneural part where you get the neural and the border region, and we can see Wnt signaling and BMP signaling playing an important role, as well as the presence of FGF and retinoic acid. You don't have to remember all the steps involved, but what you should remember is the panplacodal field is the portion that will actually give rise to the different placodes, and that occurs through the process of induction. Now, after induction, each placode will express its own unique set of transcription factors, which makes one placode different from another placode. Let us look at the induction of the otic placode as an example of induction. Now, the whole process begins when the pharyngeal endoderm secretes FGF8. Now, FGF8 is a member of the fibroblast growth factor family. So when we look at the figure, the portion in the blue box shows the endoderm secreting FGF8. The endoderm here is the lowest layer that is shown in yellow. Now the FGF8 that is secreted by the pharyngeal endoderm will induce the layer above it, which is the mesoderm, to produce FGF19 and secrete that. Once the mesoderm secretes the FGF19, that FGF19 induces the overlying neural plate to express and secrete FGF3 and Wnt family member. So thus we can see how one layer is inducing another layer to do something, which is now inducing a third layer to do something else. The FGF3 and the Wnt8C that is produced by the overlying neural plate will induce the expression of transcription factors in the neighboring cell. It specifically induced the expression of PAX6 in the neighboring cells, which will then form the otic placode. Thus, we can see how the process of induction causes the otic placode to form. Now, the vertebrate eye formation has been one of the most well-studied processes, and it results from a series of steps that involve many factors in the process of reciprocal induction. We had learned about reciprocal induction when we studied chapter 4. The lens placode gives rise to the eye, and the process by which the lens placode does that is it first comes from an eye field that is generated at the anterior end of the neural tube. So let us look at how the eye field is generated in the first place, which will then result in the lens placode formation. So noggin suppresses BMP signaling that allows the expression of OTX2. We know that noggin plays an important role in neural induction and that the neural tissues are able to form through the actions of noggin suppressing BMP signaling. Now, the accumulation of OTX2 or OX2 in the ventral head region results in the expression of a protein that's called as the ET protein. The ET protein induces the expression of a homeobox protein called Rx. Rx, on the other hand, induces the expression of PAC6. PAC6 is the same protein that we saw when we looked at the otic placode formation. Thus, when we look at the picture in the blue box, we can see that in the neural tube region, an eye field has been formed. So PAC6 activates other genes, and all the activity of those genes results in a single eye-forming field, and that is what is shown here in the blue box, and it is located at the center of the ventral forebrain. However, we all know that we have two eyes, and so for the two eyes to form, that eye field has to split into two parts. So for separation of the single eye field into its two parts, PAC6 must be suppressed. So PAC6 is involved in forming the eye field, but for the separation of the eye field, PAC6 activity is actually detrimental. This is where sonic hedgehog comes in. So sonic hedgehog that is made by the prechordal plate will suppress PAC6 and allow the eye field to divide into two different parts, which is what is shown here in the figure in the blue box. Now to look at where is this prechordal plate is present in the part of the figure that is there to the left in the blue box. So it is right below the ventral forebrain, which is where all of this action of eye development is happening. Now to look at the presence of sonic hedgehog and its importance, let us look at the figure that is there in the blue box. So here we can actually see where the eye field has separated, and we can also see the other placodes that are going to be forming. So the olfactory placodes, we can actually see them in the two smaller blue boxes. So that also requires a splitting event to happen. And we can also see the optic ones where we can observe in the other set of blue boxes, and this is where we have seen separation. This is what is normally going to happen. Now let us take an example where sonic hedgehog activity is suppressed. When its activity is suppressed, we can see that all the placodes are joined together, so we don't see that separation of left and right. And this causes cyclopia. So in cyclopia, we can see the organisms have only one eye, and there are a lot of other facial deformities. Thus, sonic hedgehog plays an important role in eye formation by separating that eye field. Let us quickly go through the different processes of eye formation and start at the gastrulation stage. So when we look at the part that is shown in the blue box, we can see where gastrulation has occurred in the embryo, and the neural plate is shown and with the alphabets NP. We can also see the preplacodal ectoderm that is shown as PPE, and the part of the preplacodal ectoderm that will become the lens ectoderm that is shown as PLE. Finally, we can also see the portion that is going to become the retina, which is PR. Now, when we look at the next stage that is shown in the blue box, we can now see that the eye field has actually undergone separation, and hence we can see now two places where the lens ectoderm can be found, which is shown in PLE. We can also see the portions which will form the retina, and we can also see where the mesoderm and the endoderm is, which are shown in pink and yellow. Now, after neural tube closure happens, and we have, by this time, we already have the eye field that has split into two parts. So these eye fields evaginate and form what are called as the optical vessels. The optical vessels are shown in the figure as OV. Now the optical vessels will secrete factors that induce the surface ectoderm to form the lens placode. And that is what is shown in the blue box, where we can see the optical vessels that are actually contacting the surface ectoderm. And this surface ectoderm that is shown as LP, which is the lens placode, it thickens, and it is what becomes the lens placode through the paracrine factors that are produced by the optical vesicle. Now we can also see the same process in the figure that is shown below. But in this case, what I want you to pay attention to is the portions that are in the blue box, where we can see in the right, the lens placode has formed and hence it is thicker. So as I had mentioned below, a placode is nothing but a thickening of the ectoderm, and this is what you can see here. Now this is called as the lens placode because this region is going to form the eye or the lens of the eye. The lens placode cells will in turn secrete FGF that results in induction of the adjacent cells of the optical vesicle to form the neural retina. So when we had seen in the previous slide, we saw that the optical vesicle cells will secrete paracrine factors that will induce the surface ectoderm to become the lens placode. Once the surface ectoderm becomes the lens placode, they will secrete paracrine factors like FGH that will result in the induction of the adjacent cells that are present in the optical vesicle to form the neural retina. And this is the example of reciprocal induction. The optical vesicle then will change its shape to form an optical cup, and the cells in the outer optical vesicle become the pigmented retina. And the lens placode cells will differentiate into lens fibers and lens epithelium. So here we have our optical cup that is present and that it actually will line the lens placode. And when we look at the figure to the extreme right, here we can actually see how the optical cup is now covering and in contact with the lens placode. Now the part of the optical cup that is right near or next to the lens placode is the one that is going to form the neural retina. The portion of the optical vessel that is on the outer end and that is in touch with the mesenchyme that is shown as MS, that will actually form the pigmented retina. For proper eye formation, the lens cells have to curve, and they're able to curve properly through the actions of Rho-generated apical constriction and Rag-generated actin polymerization. Thus, the cytoskeletal proteins play an important role in defining the shape of the lens cells. Now, the lens fiber cells also synthesize the crystalline protein, which is the transparent lens-specific proteins that allows light to pass through. The lens fiber cells will elongate and fill the lumen of the lens vesicle. So when we look at the picture in the blue box, we can see the lens fiber cells, which are in the form of the white elongated shaped cells, and they are contacting the lens vesicle. As the eye develops, we can see that the vesicle becomes smaller and smaller because the lens fibers are elongating and they're filling the lumen of the lens vesicle. The fiber cells will lose their organelles and nuclei on maturation, so by the end of the eye formation, they basically won't have many nuclei left. During development, the lens also separates from the outer ectoderm that is on covering it, and in the space that is generated between the lens and the outer ectoderm, we have corneal precursor cells that come. So corneal precursor cells from the neural crest will basically migrate into this space and they will differentiate to form the cornea. Now that we have looked at how the lens cells develop, let's look at what happens with the cells of the optic cup. Now the optic cup cells can form different types of tissues that include neural tissues, and this is formed from the neural retina that was in contact with the lens tissue. They also form the pigmented muscular tissue, which is the iris, and the ciliary body that secretes the aqueous humor fluid. Some of these structures can actually be seen in the figure that is shown in the blue box. Now, the neural retina develops into a layered area of different neuronal tribes, which is shown in the figure that is there to the right. So this kind of a layered structure is similar to what we have seen in the case of other neural tissues, like the cerebrum neural tissue or the cerebellum neural tissue. The different cell types that are formed from the neural retina are the light and color sensitive photoreceptor cells, These include the rods and the cones. They also form cell bodies of ganglion cells, as well as different types of neurons. Thus, we can see the formation of different types of cells from the optic cup. With this, we come to the end of what placodes are, and we looked at the different types of placodes as well as how eyes are formed.